half value of 25.7 nanomolar. But this channel was also remarkable in yet another respect. The dose response curve is not as steep as that of other CNG channels. CNG channels are cooperatively activated by the binding of 3 or 4 cyclic GMP molecules, and this steepness can be characterized by a Hill coefficient, and the Hill coefficient was 1, whereas for other CNG channels the Hill coefficient is between 2 and 3.5 or so. So does this suggest that the binding of a single cyclic GMP molecule opens that channel, even so there are 4 binding sites? And to study this, what we did, the binding site of such a cyclic nucleotide binding domain was known, and there is one interaction between a charged arginine residue and the charged phosphate group here, and this arginine is conserved in all 4 repeats or domains of that ion channel. And when you mutate this or neutralize this amino acid to glutamine, the binding is totally abolished. So we did all 15 mutations in all combinatorial possibilities. And there's I usually don't like to mutate proteins, living alone channel proteins, because you're doing too much bad to them. But we got a very simple answer. There are only 2 different kinds of channel. There are channels that have a wild type affinity of about 25 nanomolar, and there are channels that are 1,000-fold less sensitive. Basically, the activity is abolished. So what is the common denominator between the wild type channels, all these other mutants, and these mutants that are 1,000-fold less sensitive? Well, those that are sensitive have in repeat 3, an intact arginine, an intact cyclic nucleotide binding domain, whereas all the others, this arginine is missing. So this proves and shows that the binding of a single cyclic GMP molecule is sufficient to open these channels. Well, to summarize, binding of a single cyclic GMP molecule is sufficient to open these channels. They're non-cooperative, and cooperativity of activation would impair rather than enhance sensitivity. It has been always argued that cooperativity in ion channels is a final stage of amplification which is required for high sensitivity. Now think about it. There are 10,000 or so ion channels in this flagellum, but there are only a few cyclic GMP molecules swimming around, and the probability that 2 or 3 or 4 of these cyclic GMP molecules simultaneously bind to one and the same channel out of 10,000 is basically 0. So cooperativity of activation would impair rather than enhance sensitivity. And nature probably developed 2 kinds of CNG channels for regimes of low and high ligand concentrations. Now, there is, you would think this is a kind of chemosensation, a smelling reaction, but in fact, photoreceptors in sperm are a variation on a common signaling theme. Photoreceptors can respond to a single photon, and sperm to single molecules. There is an irreversible absorption of a photon, and there is almost irreversible binding of a molecule to sperm. Now, the messenger, both messengers are cyclic GMP. The response saturates when a photoreceptor captures about 100 photons or so, whereas the calcium response in sperm saturates when there are about 100 molecules or so bound. And there is a high amplification cascade that I cannot go into in photoreceptors, whereas there is a low amplification cascade in sperm. There is a simple reason. In sperm, the receptor and the enzymes is one and the same, whereas in photoreceptors, there's a visual pigment, rhodopsin, and there are G proteins and PD, and there is a high amplification cascade. And both cells sit at minus 40 millivolts, and they hyperpolarize on stimulation. Very unusual. These are the only 2 cells I know of that hyperpolarize. And the mechanism by which this hyperpolarization is brought about is distinct. In photoreceptors, non-selective CNG channels close on stimulation by illumination, whereas in sperm, the potassium selective CNG channels open. Now, I'm always asked, so I wanted to convince you that, in fact, sperm are like swimming photoreceptors, except that they are blind. What about mammalian sperm or human sperm? Well, that's quite another story, and I have no time to go into it. But you can read this in last year's in this manuscript that was published last year. To make a long story short, the chemoattractant in humans is progesterone. It's not a peptide, and it directly activates ion channels in this flagellum. These ion channels are called Cuthbert, and that is very unusual because it has been thought for almost 20 years that there are GPCRs involved or metabotropic signaling pathway, and that's not true. So these progesterone directly binds to the Cuthbert channels, opens it, and calcium flows into the cell. So I'm at the end of my talk, and I would like to thank you for your patience and also the people who were involved in this research. Thank you very much.
One is, do you know what caused the turn in the sperm? What protein are sensing and changing the shape of the head and the tail?
Yeah. It's a very interesting story. There is a cellular structure, that axoneme, that consists of tubulins, and the question is, what is the mechanism by which calcium ions modulate this flagella bead? And that is not yet known. There are several calcium binding proteins there, actually it's many, and they bind to different parts of the axoneme, for example, to spokes, to motor proteins. And this derivative, so how do the sperm, how do they make a calculus? How do they make d calcium over dt? Well, there's, it was recently published in Journal of Cell Biology, and we offer a model. It's a chemical calculation. So when you have two calcium binding proteins that, for example, bind to a motor protein or a spoke, and the binding of one makes this, and the binding of the other makes this, and you have a short delay between binding of one compared to the binding of the second, you make a chemical differentiation, and thereby you can explain the mathematical calculus.
The other question is, if you saturate the receptors, it would not respond any longer to the gradient.
Yeah, well, that's also a very interesting question. These sperm are exquisitely sensitive. I told you they work in the femtomolar to picomolar range. So the receptors need to have a high affinity in the picomolar range, which we in fact measured. And there are two answers to your question. First of all, we also determined the density now of these receptors. I didn't have time to go through it. And in fact, we were really astonished too. The receptor density is exquisitely high. They're basically paved with this chemoreceptor. And in fact, the density is as high as rhodopsin in the disc membrane of photoreceptors, 30,000 molecules per square micron. And the other question is, in order to operate over, they can operate over 6 orders of magnitude of concentration of the chemoattractant. So how do they escape saturation and maintain a few receptors when they're still sort of approaching the egg where the concentration is high? Well, they shift their sensitivity, and that depends on the occupancy level, and this is caused by negative cooperativity. these receptors are trimers, and they bind the chemoattractant by negative cooperativity, and thereby extend their scope of, or their dynamic range.
Thank you very much. And with this, we close the session. We thank the speakers for their excellent presentations, for working on acoustic pollution and glucose deprivation, and the audience for the stamina. There is one last